Thank you. First of all, a big thank you to the conference organizers for letting us take a couple of minutes and tell you about StemBioSys, and a big thank you to the audience for taking the next 10 or 15 minutes and listening to what we have to say about something that has become my personal passion over the last couple of years. So let me begin with an introduction to StemBioSys. We have a technology that we out-license from the University of Texas Health Science Center at San Antonio. That technology, the initial product out of that technology is something we call the high performance microenvironment, or HPME, to keep it simple. And that technology, as its initial application, is really designed to overcome a number of obstacles in developing any number of cell therapy applications, initially as a research product and then longer term, obviously, as we move to the clinic. We have completed an $8 million round of financing, Series A round. We'll be beginning our Series B round toward the end of the year. We had one lead investor from a technology or a venture fund, and then a number of individuals who were very intrigued by the technology, who were also very supportive of us as we went out and got the company off and running. One of the things I love to begin with, and I will share with you all that my wife is in the audience, and when I first looked at StemBioSys, I looked not as the CEO, but as an investor in the company. And my wife is a consultant to the financial services industry, and she said, so why are we putting money into this startup company? And so what I did is I actually put together my own thoughts around why I made the initial investment that I did into StemBioSys. And the first thing it does, as I said, is the platform, the HPME platform itself, really addresses the need for a high quantity of high quality cells. And we even heard this morning in the plenary session that continues to be an issue in deriving cell therapies, and will continue to be an issue as we go forward. The second thing I really liked about StemBioSys is we are entering the research market. We have a booth outside, and we are actively marketing the product into the research market. And we believe that without a lot of heroic assumptions about revenue growth, that piece of our business can become cash flow neutral. We've said 36 months on this slide. I think we can probably do it sooner than that. The other thing that I like, the third point up there, this is really a platform technology. And inherent in a platform technology is some hedging, because there's a number of applications that this can have and a number of things that it can do. It obviously will not work on everything, but we believe going into this that it will work in enough areas to give us a really nice return for us and for our investors. The technology itself, also, there's some intriguing results that we've gotten early on that indicate it may have some therapeutic benefits in and of itself, and I'll talk more about that later, that would obviously have significant value creation for us longer term. And then finally, we have assembled what I believe to be a world-class management team, but more importantly, and not to talk about myself, but really the board of directors that we've created is really something, and you'll see that in just a minute. The simplest way for me to describe what we have is to give you a very high-level view of how we make this. We begin with commercially purchased bone marrow MSCs. We seed them in a plastic culture dish. We add media and growth factors to that. We let the cells then expand and lay down a matrix, which you can see down in that second set of dishes down at the bottom. We then gently extract the cells, and what we have left then is our HPME. And the initial product that we have for the market will be in 6-well plates and T75 flasks. We certainly envision expanding that as we go forward, but as we've just entered the market within the last month or so, we're starting small and something that we know we can control and do well. The first question you might ask is, why are you so excited about this? Well, the first thing we've looked at is when we compared the HPME to other technologies for cell expansion, we saw more cells. That's good, but more cells isn't necessarily all there is to it. What you want to make sure is that those cells are of high quality too. And one of the measures we've come up with, and that we've seen repeatedly in the literature, is smaller cells are better. And we've consistently seen that the cells that are expanded in our matrix are in fact smaller. And then finally, we've looked at gene expression, and one particular one that we've seen repeatedly is higher expressions of SSEA4 coming off the matrix. Now, this data has been repeated across at least 4 academic institutions and a couple different commercial companies, and including inside our own lab. But the data we're showing here actually came out of the Langer-Anderson labs at MIT, and so that we've picked this as one sample of data that people have seen. I'll make a comment about patents, because obviously none of this is any good if we can't protect it. And we do have a strong set of patents both here in the US. We have several patents that have been issued, and we are continuing to actively pursue patents, again, here and abroad. So I've set the foundation and kind of told you what we have, and the real challenge, and I stepped into this role about 14 months ago, and the real question was, what are we going to do with it, and how do we maximize the value of this for ourselves and for our investors, and frankly, long-term for patients. And so what we've developed is really a 3-pronged strategic approach. So the first thing we're looking to do is to get the matrix in as many hands of many researchers' hands as we can. One, again, to drive short-term revenue for our company, but frankly, more importantly, to continue to learn and get people using it and finding out what the full capabilities of the HPME itself are. The second set of activities we've got going on, we began a year ago at this very meeting working with a set of partners to understand what the matrix could do. And we did these under a series of material transfer agreements, very simple agreements. We would supply the product, and they would tell us what they learned when they got it. We've developed about 15 of these. We've gotten data back from exactly half of those, and in every single case, the data has been promising enough around the application for people to continue working with it. The final area I mentioned, we really want to understand whether or not some of those early indications we had about therapeutic benefit are in fact real. So we have designed 2 proof of concept studies that are currently underway today, and we should have that completed by the end of the year. We'll make a decision at that point on where we go with it, but our current thinking is if one of those plays out the way we believe it will, that we could be in a phase 1 in man sometime in 2017. One other thing I want to touch upon, we viewed as very critical, is we needed to make this product, and we needed to make it consistently. And we developed, over the last 2 years, a very unique partnership with a group called BioBridge Global. They were originally a blood and tissue bank, and they've moved into a number of ancillary activities. They're a not-for-profit organization based in San Antonio, Texas. They actually built out a manufacturing suite for us, and then we also partnered with the city of San Antonio, who helped us with the funding to equip that space. And so we were able to build out a 1,000-square-foot manufacturing suite, GMP certified manufacturing space, so that we can reliably say we're producing the matrix in a consistent way. The real importance of this was we needed to demonstrate, frankly, to ourselves, to our investors, and frankly to potential customers, that we could take this matrix from the lab to a scalable manufacturing process and continue to maintain the quality that we had gotten, and we did that. The other thing about this space is it will let us meet, the space as it's built out will have us meet the revenue goals that we have for the company through the end of 2016. We'll obviously need to add on to this or build this out, but what we believe we've gotten out of this is a template, and a modular template, if you will, that becomes easily replicatable across multiple sites in the future. The next 2 slides really talk about some of those partnerships, because I think this is really kind of indicative of how we've thought about this and how we've gone out and worked with people. Our founder at the University of Texas Health Science Center is a shareholder in our company, on our board of directors. We're continuing to work with him. He's really working on some next generation applications of this and next generation versions of our product. We've worked with another local university, the University of Texas at San Antonio, who has done a number of the comparative studies that I talked about earlier. They've also helped us with some additional applications on the matrix. BioBridge Global built out the manufacturing suite for us. I mentioned them already. And then one example of where somebody's taken the matrix and done something completely different with it is at the University of Illinois at Chicago, Dr. Jose Oberhoser's team. They took the matrix and put islet cells on it and saw some really interesting results, the behavior and the reaction of the islet cells on the matrix itself, enough that we've continued with those studies, and I'm now going into an in vivo animal model to see what actually happens next. We should have the results of that study by the end of the year, but we were certainly very intrigued by what we saw, and they were as well, by what they saw the islet cells doing on the matrix, enough that we've all pushed forward with that, and we're really excited to see what happens next. You know, broadly speaking, on other collaborations, just to share with you how we thought about these, obviously we've got confidentiality agreements in place, so I can't talk about a lot of these in any depth, but to give you sort of a sense of how we thought about this, you know, we really went after high impact, make a difference groups, people who were leaders in their field, both in the commercial space and in not-for-profit institutions. And quite frankly, we saw this as a very cheap way to understand what this thing can do. And it was really interesting at our shareholder meeting, one of Dr. Chen's graduate students was in it. He wasn't aware of everything we had going on. And he said, you got to have $2 or $3 million worth of studies that you're doing with collaborators that you're not having to fund. It was done with products. So it was really a very good thing for us. And frankly, we've gotten just tremendous response from our partners in this and are really excited about, as I say, what we've seen. The collaborations, when we set out, we said we want to touch as much as we can. We've seen, as I said, MSCs, which is most of the work we've done. HSCs we've had people work with. We've seen some incredible results in the area of adipose stem cells. And we're really excited about what some of that has been. In the area of cancer cells, we've done some really interesting work with another local company in terms of being able to create a diagnostic tool. We've done work in cardiology, diabetes, neural diseases, bone repair, et cetera. So again, a full range of full suite of things as we've gone out and done these. We're continuing to work through these. Obviously, the ones that work, we will continue to pursue very aggressively, and we could envision joint ventures, joint research collaborations, more formal agreements than we have in place where we really see a real difference maker. My last slide, I mentioned the team at the beginning, and again, I can't overemphasize the strength of this because and the importance of this, obviously if you can have a great product, you can have a great idea, you can have a great concept, but if you don't have the people to execute it, it doesn't get done. So I'm fortunate to have a tremendous Chief Operating Officer in Cy Griffey, great CFO, and the board of directors that we've assembled, I think, is really incredible for a company of our size. Our chairman, Kathy Burzig, many of you may know Kathy. She was with Applied Biosciences. She was Eastman Kodak, and most recently was the CEO of KCI. We've had a number of people with pharmaceutical experience on the board, and it was interesting. We got a chuckle. Dr. Langer has his hands in many things, which is good for him, but we are really fortunate to have Bob Langer on our board. He's been obviously very, very helpful to me personally and in the board as well as we've thought about the applications of the matrix, where we go with it, what we do next, and how we begin to think about the future of the company. That's all I had. I really appreciate the time you've had. I will put in one plug. If you want to know more about us, the easiest way to do it is we do have a booth right outside this door, and myself or any of my colleagues would be happy to answer any questions you might have. Thank you.